So let's just get started. So, yeah, thanks, Catherine, again. It's really, really nice to see you today. And then, thanks for agreeing us to give a seminar. I think that would be very exciting. So before the talk, I'm gonna introduce today's speaker. Hi, everyone. Good morning. Welcome to our regular immune seminar. It's my really great honor to introduce today's distinguished speaker, Dr. Cathryn Nagler, the Bunny Food Allergy Professor at the University of Chicago. Cathryn earned her PhD from New York University and went on to serve as an assistant associate professor at Harvard Medical School. In 2009, she joined the University of Chicago as a professor, where she has continued to build an impressive body of research. And over the past 20 years, also, Cathryn's research has focused on understanding the mechanisms that govern tolerance to dietary antigens and the immune modulating effects of these antigens. And as many of you know, food allergies are a significant public health issue, and Cathryn has been at the forefront of investigating how commercial bacteria influence susceptibility to allergic responses to food. I would like to give you one exciting example. One particular groundbreaking aspect of her work involved colonizing germ-free mice with bacteria from the feces of either healthy or cow's milk allergic infants. And she discovered that the mice colonized with the microbiota from cow's milk allergic infants develop an immune responses to cow's milk allergens, while those colonized with the microbiota from healthy infants were protected. And this fascinating finding allowed her to identify a microbiota signature that differentiates cow's milk allergic and healthy individuals, both in human donors and in the colonized mice. I think Cathryn's work opens exciting possibilities, suggesting that microbiome modulating therapeutics could one day help prevent or treat a range of diseases, including obesity, diabetes, asthma, autism, and inflammatory bowel disease, which I love so much. Today, I'm thrilled to hear more about Cathryn, about her work on the recent progress in tolerance and immunity in the intestinal mucosa. Let's give her a warm welcome. Thank you, Cathryn.
Thank you so much for that great introduction. I'm delighted to be here. So I wanted to start with this slide, very old at this point, to frame my question. And what Francois Bach showed 20 years ago now is that as vaccination reduced, let me get my laser pointer, reduced the prevalence of infectious disease, vaccination and improvements in sanitation, we started to see increasing prevalence of autoimmune and allergic diseases. And this has been described in some settings in the terms of what's sometimes called the hygiene hypothesis, although the hygiene hypothesis was not actually formulated initially to explain this sort of disease prevalence. I think more recently, we and others have looked at this kind of reduction in infectious disease and increase in autoimmune and allergic disease in the context of the microbiome. So these are the microbes. I'm going to talk only about bacteria that populate our skin and all mucosal surfaces and are now known to profoundly influence our health. And there are trillions of microbes, particularly in our gut, and that's going to be my focus today. So our hypothesis has been that in the 21st century, particularly in industrialized, westernized societies, modern lifestyle factors have triggered shifts in the commensal microbiome, which have in turn promoted the prevalence of these non-communicable chronic diseases. So what are some of these lifestyle factors? Antibiotics are far and away the biggest culprit. In the US, antibiotics are often prescribed to infants during the first year of life for viral ear infections. Another big driver of microbiome composition is diet. So we've co-evolved as a human species with our microbiome over millennia. Our ancestors were not consuming highly processed foods low in dietary fiber. Nature's strategy was vaginal delivery and breastfeeding, and we've moved towards cesarean births and formula feeding. We initially lived in habitats that were in great communication with nature, which is a dominant source of our microbes. And we've moved to urban settings. As I've already mentioned, we've reduced the prevalence of infectious disease and improved sanitation and eliminated previously common parasites. And there's a growing interest in the role of environmental chemicals and how they impact the composition of the microbiome. So all of these factors together have served to reduce the diversity of the commensal microbiome overall and seem to have selectively depleted some protective populations. So I like this cartoon from Justin Sonnenberg because it says what I've just told you in a very visual way. It's not coincidence that the individual that's portrayed in dysbiosis with the microbiome because of all of these lifestyle factors is obese, because the concept is that our microbiome has co-evolved with us to serve important physiological functions. It was previously in sync with our physiology, and it is no longer in sync, and it's driving the prevalence of these non-communicable chronic diseases. So peanut allergy was not displayed on that initial graph from 20 years ago, but has skyrocketed, certainly in the US and Australia, during the period between 2005 and 2014, as shown here. People can become allergic to any food at any point in time, but 8 foods cause the most reactions, as shown on this slide. These foods can be pretty much divided into 2 groups. Milk, eggs, soy, and wheat tend to present as allergies in childhood and had previously been resolved by the time the individual became an adult, although we're seeing them increasingly maintained as allergens into adulthood. Whereas peanuts, tree nuts, fish, and shellfish are associated with lifelong and life-threatening allergic responses to food. So the question we've been trying to ask is, what is driving a generational change in food allergies? And we started this work by treating mice with a cocktail of antibiotics to reduce the complexity of their microbiome. And what you can see here, I'll show you many slides like this that have different bacterial taxa. You don't need to concern yourself with the specific taxa. I'll call out the feature I want you to take note of. So in this slide, we see fecal and ileal contents of non-treated NT mice and ABX antibiotic-treated mice. And what you can see is that the diversity is completely lost in the ABX-treated mice. They only have the only bacterial taxa detectable is Lactobacilliaceae, which seems to be resistant to this antibiotic cocktail. We do not make these mice germ-free by this antibiotic treatment. Instead, we reduce the bacterial load by about 2 logs. And only the antibiotic-treated mice have evidence of sensitization to food antigens upon repeated intragastric sensitization with peanut plus a mucosal adjuvant. So this gave us our first hint that the composition of the microbiome was regulating allergic responses to food. We went on to colonize mice with various germ-free mice with various components of fractions of the microbiota. And in these early studies, we found that bacteria in the Clostridia class are important barrier protective taxa because they induce the production of the cytokine IL-22 from innate lymphoid cells, and IL-22 regulates epithelial proliferation, the production of mucus by goblet cells, and the production of antimicrobial peptides by Paneth cells. So let's just look for a minute at what happens when there's an allergic response to food. So this is the scenario that many parents are worried about, is a little girl eating a cookie that she doesn't know contains peanuts. Normally, foods are in the small intestine are digested into peptides, but it seems that what makes those 8 foods prominent food allergens is that they all have a protein that's resistant to proteolytic degradation in the gut. So peanut allergens can get into the bloodstream and circulate around the body in an intact form that can bind to IgE and cross-link IgE on the surface of mast cells. That leads to mast cell degranulation, which elicits the symptoms that we're familiar with in allergic response. One of the mediators contained within mast cells is histamine. Those responses can range from urticaria, hives, itchy throat, vomiting, all the way to cardiovascular collapse. In both mice and men, the response that you elicit at a particular point in time is unpredictable and variable. So we wanted to ask if we could show if the bacteria in the Clostridia class are regulating the access of allergen to the systemic circulation, which is the hypothesis I've posed to you. So in this experiment, we treated mice with this cocktail of antibiotics for 7 days prior to weaning. At weaning, we administered the consortium of Clostridia or an IL-22 fusion protein, and then we challenged the mice intragastrically with a peanut extract. We then looked in the serum to detect ARA-H6, which is one of 11 ARA-H proteins, the dominant composition of peanuts. ARA-H is for the scientific name of peanut, Arachis hypogea. And this is what we found. Measuring ARA-H in serum, we saw that the non-treated mice had some ARA-H detectable in their serum 1 hour after gavage, but it returned to baseline by 3 hours. The ABX-treated mice had higher concentrations of ARA-H in the serum, which kept climbing. If we treated the antibiotic-treated mice with the consortia of Clostridia, we no longer detected ARA-H in the serum. If we treated them with an IL-22 fusion protein, we no longer detected ARA-H in serum. If we treated antibiotic-treated mice that were colonized with Clostridia with a neutralizing antibody to IL-22, we again saw ARA-H6 in the serum, suggesting to us that Clostridia-induced IL-22 is in fact necessary and sufficient to reduce intestinal permeability to food in this model. So this finding led us to propose that protection against allergic sensitization to food requires not only a dietary antigen specific FOXP3 positive regulatory T cell response, which had been the prevailing paradigm, but also a bacteria-induced barrier protective response. We showed that Clostridia are one population of bacteria that can mediate this response, and that they do so by inducing innate lymphoid cells to produce IL-22, which regulate access of dietary antigen to the systemic circulation. So given this baseline finding, we wanted to go on to do translational studies to see if we could develop microbiome modulating strategies. And in the first series of studies, we teamed up with Roberto Bernicanani at the University of Naples. He provided us with fecal samples from 20 infants that were either healthy, so they were in a vaccination clinic, or from his clinic, they were cow's milk allergic. And you can see, just looking at these pie charts, that their microbiotas were dramatically different. It was fortuitous for us that we looked at this Neapolitan clinic because Naples is not a cosmopolitan city. It's a small city with His clinic has almost no racial diversity, very little ethnic diversity. Everybody is Neapolitan, everybody is eating a similar diet. So I think that's why these results are so consistent. They wouldn't be this clear, I think, in an American clinic. And what you can see is that the healthy infants have the bacterial populations we've known about for 40 years. So in the United States, you can buy lactobacilli and bifidobacteria as common probiotic supplements that are supposed to improve health, although there's not much evidence that they do so. Both of these populations were dramatically depleted in the cow's milk allergic infants who instead had a microbiome that was more like what we would see in an adult. So in food allergy, we don't have access to intestinal samples as one would have in inflammatory bowel disease or in celiac disease, for example. So we wanted to develop a really robust model for food allergy that we could use to test our potential therapeutics. And this is what Paul has already mentioned. What we decided to do was to take fecal samples from 4 healthy infants from that Italian cohort and 4 cow's milk allergic infants and use them to colonize germ-free mice. To facilitate engraftment of the human bacteria in their murine hosts, we gave the mice the formula that the infants were consuming at the time they produced the fecal samples, reasoning that our bacteria eat what we eat, and we were providing the bacteria with the food that it had been accustomed to. We sensitized the colonized mice, in this case with the cow's milk protein, beta-lactoglobulin plus a mucosal adjuvant, over 5 weeks prior to intragastric challenge, and then euthanasia. And this is what we found. This slide represents 100 gnotobiotic mice, about a third in each group. In mice, we read out anaphylaxis as a drop in core body temperature. So what you can see here is that the germ-free mice, no bacteria at all, as well as the cow's milk allergic, we'll abbreviate it as CMA, colonized mice, had an anaphylactic drop in core body temperature. The healthy mice were completely protected. I told you earlier that anaphylactic response is caused by mast cell degranulation. A common mediator that we can detect in serum is a mast cell protease. That protease is significantly elevated in germ-free and CMA colonized mice, as one would expect, because it should vary with the drop in core body temperature. However, we only saw a BLG-specific immune response, that is BLG-specific IgE and BLG-specific IgG1 in the CMA colonized mice in blue here. So this slide then is proof of concept that there's something in the healthy microbiota that we can discover and use to treat disease. So that's what we set out to do. We began by teaming up with a bioinformaticist who divided the bacterial taxa present in the healthy and CMA colonized mice and their human infant donors, so that's D and M here, into taxa that we called protective OTUs for operational taxonomic units, or non-protective OTUs. We also took ileal samples from all of these colonized mice and looked at changes in gene expression. When we integrated those 2 data sets, we found 3 Lacnospiraceae taxa that were upregulated in the ileum of the healthy or CMA colonized mice, and we found that these protective OTUs matched to a particular bacterial taxa, Anorostipes caseae. We used the type strain of Anorostipes caseae to monocolonize germ-free mice, and we saw that by itself, it was able to protect against an anaphylactic response to sensitization and challenge with BLG. This is comparing AKCA monocolonized mice to the CMA colonized mice, as I've shown you previously, for the drop in core body temperature, release of mast cell proteases, production of BLG-specific IgE and IgG1, and type 2 cytokine production upon in vitro restimulation. So we knew that Anrostipes caseae from the literature, although there wasn't much literature on this bug at that point, was a member of the Clostridia class, which I've already told you we identified as the important barrier protective taxa that produces butyrate. And butyrate was of interest to us because we knew that it's one of the primary degradation products of dietary fiber. So dietary fibers are metabolized into short-chain fatty acids. Butyrate is one of the 3 dominant short-chain fatty acids. And for a molecule of only 4 carbons, it has really remarkable immunomodulatory capabilities, probably because these capabilities co-evolved with the microbiota's role in regulating the physiology of the host, because short-chain fatty acids are an important energy source for the function of colonocytes themselves. So we know that butyrate and other short-chain fatty acids can signal through G protein couple receptors. They are potent HDAC inhibitors, so they regulate gene expression in multiple different cell types, and they can even regulate cellular metabolism. So we were interested in doing more with butyrate because it has this potential immunomodulatory activity. But the problem with using butyrate clinically is that it has a terrible taste and smell. So it's completely impalatable. We teamed up with an engineer at the University of Chicago's Pritzker School of Molecular Engineering, Jeffrey Hubble, and he created a polymeric formulation for us that delivers butyrate to the distal part of the gastrointestinal tract where it's normally produced. So what I'm showing you here is mice that were gavaged with one of our 2 formulations that Dr. Hubble made for us. We called them But-N for butyrate micelles that were either neutrally charged or negatively charged. The neutral But-M released butyrate from its polymer backbone in the ileum very quickly and returned to baseline, indicated by the red dotted lines, by about 4 hours. We didn't see release of butyrate from the neutral But-M formulation in either the cecum or the colon. By contrast, the negatively charged But-M did not release in the ileum, but with a little bit of a lag, did release in the cecum to much higher luminal concentrations. The butyrate in the luminal content was measured by mass spec, and over a prolonged period of time. Even with this negative formulation, we didn't see elevated concentrations of butyrate in the colon. That might be because, as I mentioned, butyrate is a major energy source for colonocytes, so it could be immediately consumed. But this analysis told us that we could cover a large part of the GI tract over a prolonged period of time if we mix together the neutral and negative But-M formulations. So in the preclinical models I'm about to show you, we administered a 1-to-1 mixture of neutral and negative But-M twice a day to try to keep the concentrations of butyrate elevated around the clock. And this is what we found. So in these experiments, we have mice that were given vancomycin to create dysbiosis, to deplete the protective bacteria, and then treated, in this model, sensitized with peanut plus mucosal adjuvant. After several weeks of treatment, we challenged the mice, a group of mice, to see that the induction of an allergic response was uniform. So this is a therapeutic model, unlike the prophylactic models I've shown you in earlier slides. We found, looking at drop in core body temperature, yes, that we had induced a very uniform allergic response. So we went on to randomize the rest of the mice into 2 treatment groups. The mice were either gavaged for 2 weeks with PBS or were gavaged with the butyrate micelle mixture. And what you can see when we look at challenge is that the But-M treated mice were dramatically resistant to the drop in core body temperature when we compare them to the PBS-treated mice. Bute-M treatment significantly reduced the release of mucosal mast cell proteases, as well as plasma histamine, as well as peanut-specific IgE. So very effective at protecting against an anaphylactic response. I've been arguing to you that the benefit of our formulation is that we're delivering butyrate to the distal part of the gut, and that sodium butyrate can't do this. So we had to test sodium butyrate and Bute-M against each other, and that's what I'm showing you in this slide. So the same kind of treatment schedule, except now we have 3 different treatment groups. And what you can see here is that Bute-M is, again, highly effective. Sodium butyrate administered intragastrically at a similar butyrate concentration, no effect at all. No drop in mucosal mast cell proteases, no change in peanut-specific IgE. We also looked at the composition of the microbiota after 2 weeks of treatment with Bute-M. And we were excited to see that Bute-M treatment enriched the abundance of Clostridium cluster 14A. These are the dominant butyrate producers in the gut. And we think that the reason this happened is because from work from Andreas Baumler has shown that another immunomodulatory capability of a butyrate is that it can signal in colonocytes through PPAR gamma to shunt the colonocyte towards beta oxidation, consume oxygen, and create a hypoxic niche. So we think our butyrate treatment is creating a local hypoxic niche, which allows for the expansion of the anaerobic oxygen-sensitive butyrate-producing Clostridia in cluster 14A. So I showed you this, that we created a block copolymer formulation to which butyrate was conjugated. It self-assembles into micelles with butyrate in the hydrophobic core. We have a neutrally charged formulation and a negatively charged micellar formulation. When we administer them intragastrically, butyrate is differentially released in different parts of the GI tract. The polymer backbone is excreted in the feces. Treatment with Bute-M, butyrate is this symbol, expands populations of Clostridia, fortifies the epithelial barrier, reduces mast cell degranulation, antigen-specific IgE, and changes in antigen presenting cell populations that I haven't told you about. So it restores immune homeostasis in the gut. At the same time we were doing the initial study, the Nature Medicine report with the healthy and CMA colonized mice, another group was looking at the populations of bacterial taxa that were dominant in the infant gut. And they identified 20 bacterial taxa that were so characteristic of the infant gut that they were age discriminatory. In other words, if you knew the percentage population of a particular bacterial taxa, you could predict the infant's age. And we were very excited to see that right at the top of the list was Anorostipes. So a dominant bacterial population in the infant gut, and Anorostipes was in the same percentage that we found it at 6 months old in our infant population, so about 1%. So we decided to pursue Anorostipes caseae as another form of immunomodulation. And this is the work of another graduate student. What she decided to do was to create a synbiotic. That is to pair a caseae as a new kind of probiotic. I'd rather call it a live biotherapeutic to distinguish it from lactobacilli or bifidobacteria. And the idea of a synbiotic is that you pair the bacteria with its ideal prebiotic substrate to allow it to use that substrate to optimize growth and metabolite production in vivo. She screened a large panel of prebiotics in vitro and finally decided on lactulose as the prebiotic to use in these studies. And that was because she was able to show that Acacia forms cross-feeding relationships with Bifidobacterium in vivo, such that these primary degrader bacteria can use lactulose, metabolize it into lactate and acetate, which then butyrate producers like Acacia can then use to produce butyrate. So she started these studies by isolating a novel strain of Acacia from an infant in our cohort. And as I mentioned, it was at only 1% abundance. Sorry, my dog wants to join the Zoom. And she was able to isolate it to homogeneity and show that it was a novel strain of Acacia that we named after her, LAHUC. This new strain of Acacia produces butyrate in vitro to a level equivalent to or better than the existing type strains. So the challenge for live biotherapeutics that are of interest to many different companies is that they usually will not engraft. And the only way you can get them to engraft is by treating the recipient with antibiotics. That's undesirable because antibiotic treatment is usually part of what caused the problem in the first place. So we were very surprised and encouraged to see that AKCA was able to engraft in in this case, in cow's milk allergic microbiome colonized mice with no antibiotic treatment. And this seems to be a unique property of this, well, maybe not unique, but a special property of this bacterial taxa because we've tried many other bacterial taxa and they don't engraft. But in keeping with her idea of creating a symbiotic formulation, the engrafted Acacia did not produce any butyrate unless she also treated the mice with the prebiotic, that is lactulose, so as a synbiotic. And now when we look in the cecum, we see significantly elevated concentrations of butyrate in the symbiotic treated mice. We saw that symbiotic treatment modifies the fecal microbiome. All of our experiments are carefully litter-mate controlled. We saw an increased abundance of both the Bifidobacterium and Anorostipes itself. Of course, we administered Anorostipes, and it forms this cross-feeding relationship with Bifidobacterium in the symbiotic but treated mice, not in the CMA colonized mice. And then finally, we were able to show that the symbiotic treated mice were protected against an allergic response to food. So I'm showing you here again the CMA colonized mice, healthy colonized mice in orange, and the CMA colonized mice treated with the symbiotic. They're not exhibiting a drop in core body temperature. The symbiotic treated mice have reduced serum concentrations of mast cell proteases, reduced BLG-specific IgG1, reduced BLG-specific IgE, reduced BLG-specific IgG1. So finally, in another series of studies pursuing immunomodulatory components of Clostridia, two graduate students in the lab have decided to look at the immunotherapeutic properties of secondary bile acids. And what they've done is created an engineered live biotherapeutic. And the way that they did this was to microbiome engineer a strain of R. navis such that it can no longer make one of the hydroxylases that's required for the production of iso-DCA. So bile acids are released into the small intestine after a meal. They help with the digestion of dietary fat. Most of them are reabsorbed, but about 5% go on to the colon where they are modified. The primary bile acids are modified into secondary bile acids. Some of these secondary bile acids have been described as being potent immunomodulators in the literature. One of these is iso-DCA. A lot of the work, well, there's not a lot of work published. Of the work that is published, the microbiome engineering that was done in other strains, not Clostridia strains itself. Clostridia are the primary producers of secondary bile acids in the host, and there are very few taxa that are able to do this because they have to have they have to have each of the enzymes required for the creation of primary and secondary bile acids, and then these modifications of secondary bile acids to create iso-DCA. So what Eddie and Jack did was to pair a mutant or not mutant strain of R. navis lacking this final hydrolase with another bacterial taxa, a Clostridia P. hirinotis, that possesses both the bile salt hydrolase and the bioperin required to produce secondary bile acids, which can then be modified into modified secondary bile acids. They were able to show that they had created stable mutants of the knockout versus wild type, which you can see here. And importantly, in antibiotic-treated mice, administration of the R. navis wild type or knockout together with the P. hirinonis differentially produced bile acids, as one would expect, and only in the presence of the R. navis wild type did we see significant increases of RR gamma T positive, FOXP3 positive Tregs in the ileum, in the colon. So what they're doing now is trying to use this pair of modified Clostridia to treat inflammatory bowel disease in the CD45RB high knockout model. So in all of the data that I've shown you, we've basically broken Clostridia down into its component parts and looked at how they modulate the immune response. So I've told you about how dietary fiber is fermented into butyrate, and that butyrate can regulate mast cell degranulation to block an allergic response. It can also block antigen-specific IgE. And I didn't show you, but it can modulate the activation of antigen-presenting cells. That was the first paper. In the second paper, I told you about the creation of a symbiotic formulation of a butyrate-producing Clostridia. I didn't show you, but I'm going to tell you about it next, that we saw reduced production of IL-25 in the symbiotic-treated mice. We also saw that the symbiotic can regulate populations of regulatory and T follicular helper cells in the draining mesenteric lymph node. In a third story that I didn't have time to tell you about today, we looked at a butyrate-independent clostridial pathway, and we saw that Clostridia can produce indoles from dietary tryptophan, and those indoles signal through the aryl hydrocarbon receptor or innate lymphoid cells, which produce IL-22 to regulate barrier permeability. That's the initial story I told you about. We found that commensal flagellin, so the clostridial flagellin itself, signals through TLR5 to activate dendritic cells to produce IL-23, which is required for the ILC3 to produce IL-22. And then finally, I told you about this ongoing work about looking at how primary bile acid modification to secondary bile acids can lead to immunoregulatory secondary bile acids that can modify regulatory T cells and Th17 populations. So all of this work I've told you about so far has been focused on the protective role of clostridial products or metabolites in preventing or treating an allergic response to food. But in one of the very first slides I showed you, I told you that the CMA colonized mice have an increased response to, have an increased food antigen specific response that's promoted by the CMA microbiota. And we were interested in this work from Teresa Allengott's lab that showed that a butyrate can restrict the development or the differentiation of Tuft cells and limit their ability to act as an HDAC inhibitor to modulate type 2 immunity. So there is data in the literature that says that protist or helminth succinate can signal through intestinal cells to generate a feed-forward loop of ILC2 and IL-13 that leads to type 2 immunity. So that prompted us to switch focus a little bit and say, well, we've looked at how butyrate can protect against an allergic response. Can we see evidence for any microbial metabolites that can promote an allergic response? And in particular, one of our colleagues told us that in a big data set from an intensive care unit, he saw that butyrate was reciprocally regulated with succinate. So we looked at samples from healthy and cow's-milk allergic colonized mice from a couple of different donors, and we saw that in our setting too, high concentrations of butyrate were associated with low concentrations of succinate, and the converse was also true. And as I told you, succinate from other microbial sources can be sensed by Tuft cells such that they produce IL-25 and generate this feed-forward loop with ILC2 that promotes type 2 inflammation. So we went back to our healthy and CMA colonized mice and looked to see if we could see increased numbers of Tuft cells. So what we've done here is colonize the mice at week 3, sensitize them just 2 times. The Tuft cells are indicated by this marker DCLK1, and E-cadherin is lighting up the epithelial cells. And you can see a pretty dramatic increase in Tuft cells in the CMA colonized mice. And this is just enumerating the numbers of Tuft cells per 10 crypts per villi. We're not really sure yet whether this requires sensitization or just the microbiota. What we know so far is if we just compare healthy and CMA colonized mice that were colonized for just 1 week, we don't see any difference. But what we're doing now is looking at comparing healthy and CMA colonized mice that have been colonized for 8 weeks, as in this experiment, with no sensitization, or with not sensitization, but just with exposure to the adjuvant. So what we want to do in this system is break it down in exactly the same way we did with the Clostridia products. What is the microbial stimulus that leads to the increase in Tuft cells? And is it sensed directly by the Tuft cells? Is it sensed by some other cell type? How does it drive Tuft cell differentiation? Does butyrate block this pathway, as our colleagues suggested in that report that I just showed you? So we're looking at now a pro-allergic role for the microbiota, in addition to the allergy protective role that we've done in its whole series of studies on. So finally, I'd like to end by thanking the people I haven't already mentioned that contributed to this work, as well as our funding sources, and take some of your questions.